Today's guest, Becca Mellema, is doing her PhD from University of Utah on Immunology. Her work involves investigating the role of platelets in various disease models. Currently, she is working on IBD model. Apart from being a grad student, she is also very conscious about how science is communicated to public. So she runs a very interesting and informative blog post named Getting the Science Right, where she writes about various scientific discoveries, theories, and related scientific posts. Please check out her blog, which is linked in the description below. Being an asexual person in STEM field, she talks about how she finds out her way till now and her experiences being an LGBTQ plus communities person and her way in the community. Listen to this podcast to know more of her story, her work, her future plans, all of that, and keep listening to Franklin Speaking. So thank you so much for coming in the podcast today. I'm so thrilled to have you on there. Can you introduce
I'm so happy
yourself?
to be here.
Thank you. So can you introduce yourself with your pronouns and what you're working on right now, or anything you can add on?
Yeah. So my name is Rebecca Mellema, and my pronouns are she, her, and I am currently a third-year PhD student at the University of Utah, and I am working on platelets, which are those things that clot in your blood. And my thesis is specifically on a receptor on platelets and how that receptor interacts with a molecule called hyaluronan, and how that receptor causes the cleavage of hyaluronan. Lots of long words in there, but I look at a receptor that makes the platelet stick on something and then cut.
Okay, that's good. So how your work is coming on? Like you're on third year. Your project is for 5 years, I think.
Technically, yes. So my program, the first year is an umbrella program. So I am third year PhD, but I'm really second year in the lab.
Okay.
So I rotated actually in a neurobiology lab, which was fascinating. I got so motion sick on the microscope.
Yes.
those who work on microscopes, you guys are my heroes, because I get so motion sick.
Yeah.
And then I worked in an immunology lab, which was focusing on type 1 diabetes.
Oh.
And that work to me was fascinating. The only problem was that I was working with, so in a lot of immunology labs, we work with mice. And the mice, to me, I was working with them, and I'm like, the mice were too young.
Okay.
And it was upsetting to me just because it's like they're young. And so when I talked to the PI or the lab leader, I said, hey, is there another project I can work on that works on older mice? Okay. Unfortunately, the project I would be picking up would be that one.
Okay.
So I was going to do another rotation, and then COVID happened. Yeah, so I actually landed in the lab I'm in currently by luck, because COVID hit and my school essentially shut down, and I still had one more rotation.
Okay.
So my rotation, I was emailing PIs, and I'm like, do you want me to rotate in your lab? No, okay, because a lot of the responses were, my lab is closed, or it's like my lab's we're not doing in person right now, so we actually can't have you come in.
Oh my God.
So I was emailing PIs, and then I finally emailed a PI, and I'm like, hey, I see you study these things called platelets. I don't know what they do, but I'm interested in rotating, because I was desperate at that point, let's be honest. And the PI actually says, like, well, I'm not looking for a graduate student, but this new guy who just joined, studies inflammatory bowel disease and platelets.
Oh my.
So you can go rotate in his lab. And Aaron's lab was like a couple months old. And
it's pretty new.
I was like, yeah, he was really new. And I was just like, yes, let's do it. I don't know what platelets are, but I'll do it. Aaron and I met over Zoom.
Okay.
And he's like, yeah, we just started up the lab.
Oh my God.
Let's have you on.
That's awesome.
So
you are in his lab?
Yes, I'm in his lab because it turned out it was a really good fit. And I discovered I actually enjoy platelets a lot. I didn't think they would be that interesting. Again, I was grasping at straws. I'm like, whoever will take me. Sure, you platelet guy, sure.
That's awesome.
Yeah, and
fitting in the lab, that's the best thing. Like a good supervisor, a good lab. What else you can ask for? That's great.
Yeah, which is it's funny because like I had been trying to find like a woman PI.
Okay.
And again, by chance, I actually met Aaron.
Okay.
And so I'm like, will this work? I really want a woman mentor.
Okay.
But it's great because the lab manager, she's a badass woman in the lab, and we just actually brought on another lab technician, and she's awesome.
That's great.
So we were actually joking yesterday. We're like, we outnumber you, Aaron.
That's great because on most places, men's outnumber females. So that's great.
Yeah.
One of the very least examples where woman outnumbers. Unfortunately, but that's great.
We're small but mighty. We are a lab of 4.
It's 3
out of 4.
3 out of 4. Yeah, that's great. So, can you talk about how did you get interested in immunology on a first place?
Yeah, it is a story because I originally thought that I wanted to be a physician.
Okay.
So I spent all of my college career I'm getting ready for the MCATs, which are the tests to get into medical school.
Yes.
I was doing all of the shadowing, and then I had a year break between when I ended college and then when I would have started medical school. And I'm like, you know what? I'm going to get my EMT license, which is my emergency medical technician.
Okay.
And after I finished that, I got everything. I did the test. I got my license, and I went, you know what? I don't really like patient care. And I actually finished the MCAT and everything. Okay. I was getting ready to do my applications.
Okay.
And I just had this nope.
Revelation.
I'm like, I really want to just work on it in the medical sense, or at the bench, because I enjoy learning about it a lot more. I don't want to do patient care. So I literally about faced the day I finished my I was in Portland at the time. Okay. The day I finished my EMT final test, and I drove up to Seattle, and where I then lived with my cousin, and I found different lab opportunities. Because I had no real lab experience at that point.
Okay.
But I enjoyed being in the whole reason why I was trying to get into the medical field was because I enjoy learning about diseases.
Okay.
So going into graduate school, and even in some of the volunteer work that I did in the labs prior, I was trying to work on asthma or diseases, something in the context of inflammation.
Okay.
Mm-hmm.
Okay.
And so I wanted to work on something that had medical implications. It's this can help in a disease.
Mm-hmm.
So I didn't have to go immunology, but just based on the labs that were available, and just that just kind of where
also, if you wanted to go to medical, immunology taught most of the things that you would have. So I
Yeah.
think, yeah, that's great.
Yeah. And yeah, and again, I did try it. I did try neurobiology.
Okay.
And I did think about going like more biochemistry,
Okay.
but I will say my lab currently, we're kind of a jack of all trades lab where we work on immunology, but we're also very heavy biochemistry focused.
Oh, okay.
So
you are getting all of the like experiences all together.
yeah, I just don't have to do patient care, which
That-
I'm happy.
yeah. So you didn't
And I
want
don't-
that, and that's great.
Looking back on it, I'm happy I didn't go into medical school. I'm happy it's like you had a passion for studying diseases. We just had to redirect it.
Yes, that's great. Like you fit into right place. Great for you.
Everyone gives T cells and B cells all the credit for immunology, but platelets are really cool. Platelets need to have a lot more love.
So it's our tagline also that platelets are cool.
Yes, platelets are cool. Come on, guys. T cells, B cells, sure, but platelets are cool.
Yeah.
Because like we look at in my research, we look at in the context of inflammatory bowel disease or IBD, we actually look at a extracellular matrix molecule or ECM molecule called hyaluronin.
Okay.
And we look at how platelets degrade hyaluronin in a disease. Okay. So in a healthy individual, hyaluronin looks like a nicely mowed lawn. I know I'm going to get to platelets in a second. I know I'm talking about this new weird molecule called hyaluronin, but bear with me. So a healthy person, nice mowed lawn looking hyaluronin. But when you get sick, at the site of inflammation, the hyaluronin will grow and it will look like, out of control growing lawn.
Yeah.
Exactly. And so that hyaluronin can actually act as landing sites for a lot of immune cells. It can actually, it can let things land on it, like infiltrating neutrophils.
Yeah.
Exactly. But and again, like a relatively healthy individual, when you're getting better, you need a way to degrade that hyaluronin. Otherwise, you're going to get inappropriate recruitment of these immune cells. And that's where our study comes in, where we look at platelets that not only use hyaluronin as a place to bind and start clotting, because platelets are little clotting enucleated factors, but they also will so what we look at though is not only can this hyaluronin act as a place for landing, but the platelets also actually degrade hyaluronin.
Okay.
So yeah, and so platelets can actually have a receptor that cuts the hyaluronin and then essentially cleaves hyaluronin in theory to like a more manageable. The fragments do other stuff in your body, but that's not really what I go over in my research. Because what we're looking at in IBD, again, inflammatory bowel disease, people with IBD have we found that they actually have a loss of a hyaluronin receptor on their platelets. Called lalin.
Okay.
There's a significant loss of this HA receptor, and we think that this loss of HA receptor actually is what causes the loss of HA degradation, and the loss of that degradation then causes the more infiltration of immune cells, therefore chronic inflammation.
Okay.
Exactly. The only problem is no one knows what lalin does in platelets. It's like we know it's an HA receptor,
and that's it.
Pretty much. I mean, we know it binds to this molecule called talin in the inside of the cell, but like what I mean, we know what talin does, but like how lalin interacts with talin.
Okay, the downstream signaling pathways, and so you don't know yet.
Yeah, we don't know, and so that's a lot. That's my research.
That's very fascinating, you know.
Yeah.
Actually, it is true that immunology is more based on T cells and B cells, and what you're talking about is also like seemingly very important for the inflammation. So you were talking about IBD. So are there any other diseases where this scenario also happens? Do you know about any?
So we are going to be looking at other diseases. I'm a little nervous to go beyond IBD, unfortunately, just because I only focus on IBD.
Okay.
And I'm a little nervous to say like, I'm pretty sure it happens the same way, but like in asthma patients, we don't know the upregulation of lalin.
Okay.
Yeah. So my lab before I joined, they were looking at why are platelets dysregulated in IBD, and they found this loss of this HA receptor. And so they're like, okay, maybe this is like one of the factors that causes. Yeah, because platelets are hyperactive in IBD and cause clots in your bloodstream. And so my PI thought maybe this is like one of the factors that causes it to these platelets become hyperactive, because not only do you lose the loss of hyaluronan degradation, you also maybe have a lot of other downstream factors that are completely out of control, because you have a down regulation of lalin.
Yes.
But again, no one really knows exactly what it does in platelets. So
it's pretty like it's completely new discovery you are working on. That's very fascinating. It's great.
Yeah, because lalin has been looked at in like T cells and kind of in other cells, but again, in platelets, we're like, So I've been doing a lot of aggregation assays, where I look at, if I block lalin with an antibody, how well does it aggregate? How well does it activate? We just got our lalin knockout mice just started breeding again. Yay!
Oh, congratulations.
Yeah, I know, right?
Yeah.
And so we're actually looking at, it's like, okay, but if you have a complete loss of lalin in these mouse platelets, what does that do with aggregation? So lots of aggregation, lots of Western blots right now.
Okay, good luck with that.
Oh, Western blots. We love to hate them. When they work, they work.
Yeah.
When they don't work, they're so terrible. I just spent 2 days.
Yeah. And like when you look at the results, if that doesn't work, it's like, what did I do wrong? Like, why was I wrong?
I know. Why? Where are you?
Exactly.
Or like I just did a Western blot the other day, and like 2 of my lanes have nothing in there. I'm like, I know there's stuff in there. Where are you? I'm like, where is this protein?
Yeah. So the next thing I wanted to know is, like, you talked about IBD. So do you have any models, or in vitro, in vivo, what you are working on?
Yeah. So I work on, I can actually get platelets from both healthy donor humans, and we also have a collaboration with a lab where we can get platelets from IBD patients.
Oh, okay.
So, yeah, so I've been doing some aggregation and Western blot on that. So we hopefully are going to be able to start looking at like proximity ligation, where we label lalin and we can label another receptor we think it might be close to. And when our labeled receptors come close together, it'll create a signal, which will be like, yes, they are close together.
That's very interesting.
It's very interesting, and I hope it works because we haven't actually done it yet, but I hope it works. Because it'll be interesting to see what other receptors
are
Leyland
interacting with Leyland. Okay.
Yeah, exactly. Because again, we don't know what it does. Does it form a complex? Is it just out there by itself?
Okay.
So, and so that's not really a model. That's just me. That's just me taking blood from humans.
Humans, yeah.
But I do, so I have my mouse model though. So I know I talked about my Leyland knockout mice. And what we're going to do is that we're starting to do something called DSS colitis model, where we put DSS, a molecule called DSS, into the drinking water, and that will cause inflammation in the mouse's gut. That's very akin to colitis, which is a subset of IBD. And so what that'll allow us to do is that in both a wild type mouse versus a Leyland knockout mouse, how does that change the like the colon length? How does that change the colon morphology? How do the platelets react? That's really my big question. That said, my Leyland knockout mice are what we call systemic knockout, which means all of the cells don't have Leyland.
Okay.
Which is not what we see in actually IBD patients. We're looking at Leyland downregulation of just on the platelets. Okay. So what I'm planning on doing is that once we get our colonies back up, I can actually I can take a wild type mouse and I can deplete its platelets by injecting an antibody.
Deplete the whole platelets, not just Leyland.
Oh yeah, so what I'm going to do is that for a wild type mouse, I inject it with a specific antibody, and I can deplete all of the platelets in the blood.
Oh wow!
So the entire mouse is still intact, like it still has Leyland everywhere else in the body.
Just
Leyland isn't.
Okay.
Yeah, and then I can actually replace it with Leyland knockout platelets from my Leyland knockout mice.
Oh wow, that's
so
pretty, yeah.
And then once we have that, and once I master that, then we can put that mouse on a DSS colitis model, which we think will be closer to what we see in humans.
IBD. Okay. Okay.
So
great.
Again, so you have Leyland, because Leyland is not just expressed in platelets, but you have Leyland everywhere else in the body except for platelets. And what happens when you inflame the gut, and then does that recapitulate what we see in colitis?
Okay.
So it's kind of like building a couple of different models into one.
And see what happens. Yeah.
Yeah.
So you talked about no.
Oh no, sorry. Granted, that's still in hypothetical land because we still have to get our colonies up. So.
Yeah. You'll get there. Like I think science starts with hypothesis all the time, and then it goes further. So I think you'll get there.
Yeah. So that's
and then
working on it.
Yeah. And platelets are cool.
Yes, exactly.
Yeah. Just exactly. This is the first time I am interested in immunology, and this is the first time I am getting interested and excited about platelets. I haven't thought about this, so go platelets.
They're so cool.
Yeah.
They also keep you from like bleeding out. That's really important. I was talking to someone in the class I'm TAing in, and I'm like, they always talk about all the B cells. Show me some platelet love.
Yeah. I'm sure that you will be the trailblazer to making platelets cool.
Yeah, we're just gonna every time I go up for a conference now, I'm just gonna be like, okay, what you need to take away from this is that platelets are the bomb. Forget about what you know about B cells and T cells.
So the next thing, like you talked that you are in a new lab, and also you are looking into this very new receptor. So I am sure that you are not finding many references in your literature according to in terms of Ellen. So how is that difficult for you, or is it easier for you?
So it's a struggle because there are, again, like it's been looked at in other cell types just a little bit. And so what that does is that it's like in endothelial cells, it's like, oh, we think it might be this downstream pathway. And you're like, well, it might be that in platelets. So it gives us a starting point, but it's one of those things where in science you're like, I don't want there to be a ton of literature on the thing I am, but I also want enough that I can have like some sort of skeleton of where I'm going.
Yes.
Because I mean, because it's like, it's kind of a shot in the dark if you don't know
any different. Yeah, exactly.
Yeah, exactly. It's like somewhere it's I don't know what I'm looking at. I don't know what my downstream things are. I don't know. So, but I will say it's like even the small amount of literature still gives us some place to look at.
Yes.
So, is it frustrating? Yeah, it would be nice to know what Leyland's crystal structure is, and that just doesn't exist. It would be nice to know like what other things we could be interacting with, so that I can, without having to do all my screens. And it would be nice to just be like, I don't need to look at that. I don't need to look at that. So there's a lot more screening looking at it, especially in platelets, because not a lot of literature on that.
Yes. Yeah, I got that. It's like, it goes, I think, both ways. Like you are the one that will be looking at, and your paper will be looked at as a reference for platelets, and you are having the problem. So it's, silver lining, I can say.
Yeah, I mean, trailblazing is a good word for it. It's all my hard work will go somewhere, so someone else down the line doesn't have to do the screens.
Yes, exactly.
Future person working on Leyland and platelets, you're welcome.
So, like, going forward, what do you want to do, like after PhD, or what will be your future plan?
Yeah, so I'm bouncing between two ideas right now. I'm either going to go into industry or I'm going to go into science communication.
Okay.
And I've been talking with my PI about it quite often.
Okay.
Because I really do enjoy talking about my science and getting it out there, maybe doing policy. But I'm also, it's like maybe I just want to, be a lab technician in a industry job. And what I do know is I'm not really into the academic. I kind of want to go a non-traditional route.
Yeah, that's awesome. Like, do whatever interests you.
Yeah.
That's great.
I think mainly because I don't want to be a PI.
Okay.
I don't want to do my own lab.
Okay.
Not for me.
That's like, everyone has their choices. So yours, whatever it is, either science communication or your lab technician, it's your call.
Yeah.
And that is like interesting you brought it up, because I think students, those are in PhD, doing PhD or in this kind of field, has the kind of pressure that they have to go to academic, like have to do postdoc and then whatnot. So the similar kind of conversation I had with one of my lab mates, she doesn't want to go for like becoming a PI or anything. She wants to be a professor just after doing her PhD. That's tough, because most of the professor either has some kind of postdoc or RA, experience. So yeah, that's a great thing. So what do you think about that? So like, how can we change the, perspective of people going into academia? They do not have to be in there.
Yeah. So
Sorry.
I'm actually on a committee right now. That brings in speakers to our department. Or general, it's a slew of departments, but we've actually been talking about it this year, and we're helping out with this thing called Career Day. So we're bringing in non-traditional, we're not bringing in non-traditional jobs. So that's how we have been trying to, I'm not gonna say combat it, that's the worst thing, but like we're trying to bring light to like there are other options besides academia. Because like, hey, there's someone who works on wow, policy. I'm doing a terrible job. It's 9 o'clock my time, guys. I apologize.
Yes. No, that's fine. That's fine. Yeah, it's very important that we talk about alternative lines also, because there are different sets of fields that people can go to, and they're not just aware of the things.
Yeah, and what I have also found is that the PIs need to be open to that as well, or any person in charge of a lab, because I did have a conversation with someone who I could have potentially joined.
Okay.
And when I said, it's like, I'm not thinking about going into academia, they're like, are you sure? Are you sure though? And you're like, oh, okay, back up. If I have a gut feeling that academia is not where I want to go, I'm being upfront about that.
Yes.
And so I think even if the PI doesn't, like, I don't think that's the route you need to go, you still need to be able to go, it's like, okay, how can I get you there though?
Okay.
It's not your path. It's not like the PI's path. It's
your
the student's path. Yeah. Yes. Student or postdoc or whoever is asking for it. Because for all you know, it could be a startup or something. It's like, I'm
definitely.
And I think so like we were talking about while we were just chatting before this about like science communication issues with like the coronavirus situation. It's like it's pretty clear. It's like we need people to go and be invested in science communication.
Yes, definitely.
So it's like, so that's a weak link that we need to work on. It's like, and that would be something that a PI should be like, yes, you need to go into science communication because we clearly need it.
Yeah, definitely. We need this, like, science communication more than ever, because like the general public is not very aware of like what is going on behind the scene, like in science, and there has been some misconceptions. So definitely, it's very
and like
true.
And another thing that we as scientists need to get better at is actually communicating. Because if we just like random scientists who doesn't actually do practice science communication explain this treatment, we're not well trained in, at least for my PhD program, to like actually speak to anyone outside of our field. And so like when I was
academic language also, if you talk about all hyaluronic and all of that stuff, I think most people like it's not normal to understand, like they wouldn't. So in a layman language, it's very important.
Yeah, and so I usually have to have a translation for what I do. It's like I look at a receptor that binds to a thing that cuts it.
You said it.
That is what you need. Yeah, it's like that's what you need to know, though. It's like like to know what I do. It's like you don't need to know what hyaluronate is or what hyaluronate does if you're just trying to be for how platelets regulate the homeostatic conditions of hyaluronate. No one's gonna understand that. Even people in my field will also look at me like, deer in the headlights, because like, what are you talking about? I don't work on that molecule. What are you doing?
Yeah, it's obviously, if you talk about like someone from like say plant biology, they would know the name, but all of the detailed stuff they wouldn't understand, obviously not.
Yeah, and so it's like
it's vice versa.
Even in our own departmental seminars, we're like, I don't understand what's going on. It's because they assume I understand what you're talking about. We just need a like, here's what diabetes is. Just like the beginning of your talks. Just like brief overview of what for the fellow scientists out there, just a brief layman overview before you go diving into your work.
Yes.
Please. Many a PhD student will bow to you and be so grateful.
It's nice you brought it up, because my PI, my supervisor, always tells that when you are giving a presentation, always think that your audience doesn't know anything. So if you are like going to go like T cells or like TC cells or whatever the acronym you are using, just tell what it is, so that the whole audience will understand what it is. So she does a great job in presentation. I am like learning. The other thing I wanted to talk about, you were featured on 500 queer scientists. So, and you talked about a little bit about science, about your orientation. Do you want to talk about like more about yourself?
Yeah.
Were, yeah, talk about it.
Yeah. So I identify as asexual and aromantic. Um, cue the mitosis meiosis jokes for asexuality. We've heard it before. It's also a joke in the community, but so I identify as asexual because I have no attraction to anyone. I don't think anyone looks hot. I have no desire to sleep with anyone. I am someone who I also have no desire to kiss anyone. That's not the case for a lot of asexuals. I happen to be, I really don't want sex. I'd rather, eat hot lava than have sex.
Okay.
But again, I want to stress that is not the case for most of the asexual community, because like you can still enjoy having sex and just like not
into it for
not desire. Yeah.
It's it's a spectrum. So like obviously not everyone is going to be on a specific wavelength. So that's what we need to understand, of course.
Yeah. And so but yeah, I can tell you, it's like I can look at a naked human body, doesn't matter male, female, and I'm like, meh. I don't get it. I don't like, every time there's a sex scene in a movie, I'm like, and I'll be on my phone. Oh, it's done. Okay. I don't get it. I mean, I get it. I grew up in a society where it's like we make sex a huge deal. So it's like, yes, I obviously get it, but I also, it's the equivalent of me. Yeah, it's like, I don't want it.
Yes.
It's like everyone wants the chocolate cake.
Yes.
And I'm like, no, I'm not hungry.
You don't want chocolate cake?
No.
You don't like chocolate cake?
Okay, wait, hold on. We need to back up. I made a very extreme example. To be clear, I love chocolate and I love cake.
Yeah, that's what I.
Okay. Oh wait, no. No. To be very clear, chocolate is wonderful. Before anyone listening to this, don't get on me for not liking chocolate. I love chocolate, specifically 60 to 70%, but
yeah.
But I was the analogy is like, there's something everyone else is like, this is so good, and you're like, yeah, no, meh. Yeah, and then and then for some asexuals are like, once like they eat it and they're like, it's good, but I'm it's like, I'm not really gonna hunt for it.
Yeah. Yeah, and that's fine. I think it's very important that you talk about it, like we talk about it more, of all the like spectrum, that it's very important to feel like we do belong. It's nothing like less or nothing like alien concept. We are here, and as you said, like go LGBTQ plus community. So that's really
it's so interesting you say that, though, because the tagline for the asexual community is, we are valid, we exist. Wow. Which I think that says a lot about how the queer community views this as a whole. Where I actually didn't consider myself queer until the middle of 2020, where I actually met another asexual for the first time, because I grew up, didn't know that was a thing. Even in college, it's like, I had a lot of queer friends, but I'm like, I'm a really good ally. Didn't quite pick up on like, I'm not interested in anyone. I'm just like, I must be default heterosexual. But that's not my thing.
Yes.
It wasn't until this is so embarrassing, but it wasn't until I did a BuzzFeed, like, quiz yourself on your sexuality, and I'm like, what the hell is asexual, once I got it.
Yes.
And And then that night when I was reading stuff about asexuality, I'm like, oh my gosh, this makes so much sense. I then made the mistake of going on to a forum asking, I didn't ask the question had already been asked, where it was, are asexuals, part of the LGBTQ plus community? And there was so much like, no, they're not. And I'm like, oh, even though A is for asexual, there's just like, no, because you don't have, like, there's no prejudice against it, or it's like it's like, no, you're really either like you're gay or you're
That's just awful.
You're actually you're heterosexual. I mean, honestly, some of the stuff that comes out of it mirrors the like bi erasure that comes out. And like we actually to give you even more of a like overview of excuse me, of like this fear of like ace phobia. I don't know what it is, but like 2 weeks ago, I think at this point, I'm bad with dates, I apologize, but we had Asexuality week.
Okay.
And people around the world were celebrating asexuality. And one of my favorite and most prominent activists, Yasmin Benoit, she's amazing. For those listening, please look her up. She's done so much for the asexual community.
Okay.
But she is a model, and so she posted a asexual lingerie line, which is totally appropriate because we also wear, I don't, but asexuals can wear lingerie. It doesn't stop us from wanting to wear. It's like, but the amount of
see.
Yeah, and the amount of harassment that she got just for posting it, and they're like, you can't be asexual if you wear lingerie. Like I was watching it just through Twitter, and like it was terrifying and horrifying.
Obviously.
And just like, and so it's like, it's one of those things where, oh, you don't get prejudice, but it's like, but we don't get the same kind of prejudice. We get infantilized, we get I can't tell you how many times I've been like, oh, you just haven't found the right person.
Oh my God. Enough of
and this whole like, and they're like, never happening. I mean, I'm also a romantic, so I was like, I'm not even looking for a romantic partner. That just doesn't, I am all for platonic relationships. Be my best friend. Don't want to kiss you, don't want to hug you, no interest in sex and no interest in any romance. But it's just a side note, it's key to note that romantic attraction and sexual attraction are two very different things. Yeah. But yeah, so it's like I get it. I have it's very subtle, but it's like, oh, you'll find someone, and the tone changes. It's like they talk to a child. They just don't believe you when you say no. It's like, I'm really not interested in anyone. And like, I don't even say to people I'm asexual. I just, it's like, under it's like, it's none of their business if it hasn't come up. It's like, I'm not going to just say I'm not finding anyone because I'm asexual. It's like, no, I'm just not looking for anyone.
Exactly.
I'm not going to get married. Or, I think this is probably the most open I have been, because like up until this point, I think just a couple of my friends know that I'm asexual, because it's not, I don't just scream it out loud, because it's like, not anyone else's business.
Yes, definitely. But I think that once people know, they should respect. I think that's the like least thing as a human being people can do.
Yeah.
And that is like very missing from, like, once you tell people how do you identify with, or how do you want them to look at you, and they just don't get it. They all want to stereotype you according to themselves, like their understanding.
I also wish that sex wasn't always linked to my value as being a woman.
Yeah.
Which is another thing where you're like, you have to have sex. Although if you have sex, then you're considered, promiscuous. But if you're a virgin, which I am, it's a, why do we use the word for, I haven't had sex. That doesn't make me pure. It doesn't make me better than anyone. It's like, it's because I literally don't want it.
Yes. It's very bizarre how this, society works, I think. It's,
yeah.
You do it, whatever everyone gonna judge for one thing or the, you do this, people are gonna judge, you don't, people are gonna judge. So that
it's just, it's like, so why do you care what I don't do in the bedroom?
Yes, exactly.
Again, I'm coming at this from like, I don't even preface that I'm asexual. I just go like, no. And
yes.
And I will say it's not from everyone. This is not to be like everyone in society. It's like, no, I definitely meet folks who are very like, yes, I will respect you. Not always though. This is for the not always people. You're like, yeah, no, no, I don't like, sorry, I'm gonna be a little graphic here. I don't like tampons in me. Why do I want anything else? I don't like being cuddled. Why on earth would I want anyone else's male or female's body on top? No.
We just don't. You just don't, and that's fine.
No.
Exactly. Yeah.
But
so
again,
So I was wondering that, have you come out to your work friends or your colleagues?
I've come out to one of them.
Okay.
I'm going to be vague on who I have, just so that I,
Yes, definitely.
So it's like, I haven't come out officially to my PI, but
okay.
I do know he is very aware of asexuality, and so it just hasn't come up.
Yeah.
In fact, I might send this to him and be like, "Hello, Aaron. This is how you're finding out."
So the question was like, how do you navigate through all of this, like academia and your sexuality? How do you navigate through this? Is there any hardships, or have you seen any positive responses?
So to the folks that I have come out to, it's very positive response.
Okay.
That said, I wish I was more open about it, and I'm always just on the cusp of like, maybe my lab is, but I don't know, if is the entire floor open. It's weird because like a lot of people would just go like, what is that? And my fear is that it's not that there will be outright like, we don't allow asexual. It's like, that's not what's going to happen. My fear is that they're going to say, oh no, you're not, or something along the lines, or it's like, oh, you just haven't found the right person yet. And so, and it's like, so there's a degree of me not wanting to explain it to every single person, because that's just going to get tiring, because it's like, not everyone knows what asexuality is. And also, I only know one other ace person. I only ever met technically two. So haven't met her face to face. That's why it's like we've met over Zoom.
Okay.
So I'm going to say two.
Yeah.
But physically one, and like, that's intimidating, where you're like, I only physically know in my building your campus, or it's like one other asexual
person.
Yeah, and that's scary.
Yes.
And it's like, that's where I'm at right now. Is if it comes up, it's like I will tell, but I wish I was more, but I'm also just not going to be like, yeah, walking down the hallways, I'm an asexual. So there's.
So I think there is like I have just one, other than you, I just know one asexual person. So she also talked about like, I feel that queer people are also not that welcoming, or encompass that asexual person, but I think that the plus sign and A tells for themselves. Like we are all in the umbrella. So whatever you like identify with, you are here.
Which I wish that wasn't our tagline, though. I wish it was just I don't look for. It's like I'm not interested in sex or something like that. Instead of like we need to validate our existence because the majority of our responses is invalidating our existence.
Yes.
And so I would for those who are listening, it's like keep in mind that is our tagline. We are valid. We exist. Yes. There's a reason that is our tagline. So it's like think about why that tagline exists.
Yes, definitely. Like I can relate because like I don't know anyone physically who is gay, like in my place. Anyone, not a one person. Like not in scientific community, not in anyone other. Like I just talk to like people on Instagram who are also queer. That's it. So it's very jarring for me to know that, because it's India. Obviously there are people who are like women are fighting for like their freedom, so it's bizarre to ask for like to understand that what is LGBTQI plus community is. I cannot ask for that because I know that people would understand. So that's very, very struggling for me. I do feel that I just wanted to share that.
For those who have ever read 1984, there is like you are a minority of one. And that is such a powerful line where that's what it can feel like. Because it's, again, like I interacted via like Twitter and Instagram.
Yeah. So, talking more about LGBTQ plus community, could it be helpful when we are going to conferences or like we are in a like a bulk, we are in a room full of people, if we know that those communities are allies or positive about LGBTQ plus, do you think that talking about LGBTQ plus would help that scenario? Am I clear?
Yeah, it just so I'm just making, so it's more just like if we just talked about it a little bit more or made spaces that were much more open, which you're kind of seeing right now. I mean, it is like knowing that there is an LGBTQ community within the STEM community specifically is I mean, coming from someone who up in like very recently determined that she was queer, even before that, it was still very like reassuring that there was an LGBTQ plus community.
Okay.
Mainly because I also have a bunch of friends in there as well, and so I mean, yeah, I mean, here's the thing: representation is always going to matter because otherwise, you're going to think that you're like, if in the case, if you're like me, if you're like in the case of an asexual, for a long time, I never met anyone else. I'm sure there are not just asexuals, but like anyone else out there. I don't think I'm doing a very good job answering this question, mainly because I just very recently determined that I was LGBTQ. I was queer, and up until a couple, like maybe a year ago, I thought I was just on the periphery of it.
Okay.
So I am still trying to look at that and going like I am within that community. I still struggle with like, oh right, I am queer.
Okay.
I can be in the LGBTQ plus community as well. So that's why that question is a little difficult for me to answer. Not because
that's like, fine. This is also important that you said that like you find out recently and like your story. That's what we are asking for. Like that's what I am asking for. That's all. So okay. So what are your stories? Yeah.
So a couple of hobbies of mine. I am a cyclist. I love my road cycling. I used to race in college, and then I've gotten into mountain biking and cyclocross,
Wow.
and I've dabbled a little bit in track racing.
Wow.
Just a tiny bit. I actually had free lessons at one point. I don't remember how I got those, but
So you know all, like you know all about cycling. You were all places about cycling. Yeah, that's great.
Oh yeah. I also love to do my own mechanic stuff. So once my parts get here, actually this next week, I'll be working on my bike.
Oh, that's great.
Plug in some tunes and take it apart and clean it.
Oh wow, that's great. You have very mechanical hobbies.
And what else do I do? I do play. I play Dungeons and Dragons. That's another one of my loves.
Okay.
So I've played that since I was 10, and
so you're a champion.
What's sure, yeah. It's a never-ending game. It's just like a long storytelling game with like every once in a while you have combat thrown in there.
Okay.
And I also love creative writing. I would love to publish a short story someday.
Do it. Yeah.
Yeah. Well, we're trying. I keep putting out there to magazines, and I've gotten a lot of rejections. But one day.
Yeah, you will get there. You know, you talked about you have also a blog post, I think.
Yeah, I do have a blog where it's called gettingthescienceright.com. Right as in WRITE.
Yes.
And what I do is that I have two different types of posts. One of them is looking at science and how you can accurately use science in your stories, such as viruses. Exactly. So it's like, hey, this is how viruses are used in science stories. This is what they get wrong, and this is what you can do to get them right.
Okay.
And then during the pandemic, because I think everyone reached that mode of like, I've gone crazy, I've gone stir crazy.
Yes.
I decided to start making blog posts about like world building, how to make scientifically accurate
stories.
Not even stories, but like creatures. So like I did a post on Medusa, how to make a possibly scientifically accurate Medusa. Mermaids. I actually made a post about how you can look at mermaids. It's like, do you look at them as a fish or do you look at them as a mammal? So they're two different things.
Yeah. That's you are connecting scientific communication and creative writing together. It's very fascinating. I would link your blog post on the show note. Definitely.
Yeah.
Okay.
And I'm currently writing a blog post right now about like venoms and like all the different cool molecular things that happen in venoms. Really cool. Fun fact: bee venom actually has one of my favorite molecules, hyaluronidase, which cuts the hyaluronan so that the venom can so that the venom can go through the like skin layer easier and the epithelial layer.
My god!
Yeah. So it destroys the ECM and allows the venom to like go farther.
Oh yeah. Oh my god.
So but that's still in the works right now. And then I'm trying to think of what my last hobby was that I mentioned. I mean science, but that's not really a hobby.
That's kind of a profession.
I know, right? Yeah. I do science for a hobby, 12 hours a day kind of thing.
Yeah. So the next question would be like, what would be your advice to people who are, in the transition that you talked, that they wanted to be in a like a disease related field, but they don't want the patient care. And also people from, I think that, the diverse set of people. What are your advices?
So for those who are, those who are just looking to like go maybe into a PhD and just getting into grad school, that's how I'll interpret that. B, I would say, one, it's okay to get rejected, because I got rejected my first go around, so it will happen. Maybe you're one of those lucky people who get in the first time, and I salute you. If you're not, it'll be okay. I got it in my second round. For those who start to go into your PhD, and you have an opportunity to like check out different labs, don't be afraid to maybe, if you find that this is not your passion, because you do change a lot in graduate school. Like you're put into a pressure cooker, and so you will change. Yes. And you may find that what you thought you were going to go into graduate school for, that may not be what you're like, what is actually. Yeah. And so don't be afraid to like maybe try out other things, and I would say don't panic if you find that what you thought was your passion is not your passion. Yeah. Because I thought my passion was going to be, being a doctor, and nope. But I would say like don't be afraid to maybe try a different path. And I'm talking about it obviously from my life choices, going from getting an MD possibly and then shifting to getting a PhD. But I would still say it still applies to within a PhD. Okay. Because if the lab is just not working for you, but you still love research, try like maybe look at different mentors and whatnot. For those who are like, for diverse people, I actually meant like people from the LGBTQ plus community. Yeah. Okay. So we were actually just talking about labels, so don't worry, there is a label for who you are. There's so much. For those who are already well established in the LGBTQ community, and who are willing to be open, it's so important, and we do salute you, because those of us who like I was questioning for quite a while, and those who are willing to be approached and show that they are part of the LGBTQ plus community, like that is a huge deal for us to see, one, that we might be able to see ourselves in you, and that it's also that we exist in the STEM community. I know what I just said was more towards folks who are already out in the LGBTQ plus community, but those who are still looking, there are resources, and keep looking. You will find someone. It might be by chance, or it might be because you found, you went to a specifically an LGBTQ plus community. Also, if you are asexual or aromantic, you do belong in the queer community. So that if you do identify as that, you are not on the edge like I thought for years. You are in the queer community. So it's an umbrella. So everyone is there. A does not stand for ally. It stands for asexual, aromantic spectrum. So, because again, Sheila, I'm gonna plug if you would like to learn more about asexuality and aromanticism, anything on the spectrum, on that spectrum. Let me try that again. I'm so sorry. If you would like to learn more about like asexuality and the aromantic spectrum, I would recommend looking at Yasmin Benoit. She is a major asexual activist, and she has done amazing stuff for the asexual community, and I would highly recommend you check her out. even if you're not questioning if you are asexual, please check her out because her stuff is amazing.
Okay. So, send me, you already have sent me those. I will link that up. Okay.
Yeah.
And thank you for the recommendation. I'm sure that people who are questioning would be, find it helpful. Definitely. So I think that's it. Do you have anything else to talk about?
Platelets are cool.
Yeah, platelets are cool.
I'm gonna be the person who yells that because I'm the only one who's gonna be doing it. Platelets are cool.
That's like, I keep saying that, but I think that's so, what can I say, fulfilling that you are kind of the only person that's fully working on platelets. And
I mean, I am in a group that works with platelets. I just wanted to point that out. But you have given me too much power. But too much power, and I will stand on this hill.
If you just search T cells on NCBI platform or anyone, there is thousands of paper like T cells for but for platelets, I think I have heard it for the first time, and I am feeling very fascinated by it, and I'm sure that people who would listen to it would definitely find it fascinating.
Mammals are the only ones with platelets, by the way. Fun fact.
Okay, yeah, fun fact.
Other animals don't have platelets. They have stuff like thrombocytes.
Yes.
But we have platelets.
Yeah. And they're cool.
They're cool.
Okay, that's
too much power.
If you want to take away from this podcast, it's platelets are cool. Yeah. So thank you so much for joining in the podcast. And I want to point out that for the first time we did the podcast, it ended very abruptly due to some internet problems. And very generously, you talked with me and also offered me to do a second time. So I'm very, very grateful for that. And I thoroughly enjoyed this conversation. I personally get to get so many insights about platelets and that they're cool. And so I would definitely now check out more platelet related papers. So thank you so much for being in this podcast, and thank you.
Yeah, no, thank you for having me on twice.
Thank you so much.